Hello everyone, this is Razan from Pendleton, Oregon. Today I'm going to talk about soil organic carbon dynamics in Pendleton tillage fertility long-term experiment. Oregon State University Columbia Basin Agricultural Research Center have several long-term experiments established in the past. These long-term experiments serve as a valuable resource for Eastern Oregon producers to understand the impact of agricultural management practices on soil quality and crop production. Studies at Pendleton long-term experiment revealed nearly 50% soil organic carbon loss from 0 to 60 cm soil depth in winter wheat summer fallow system. When we looked at soil organic carbon in surface soil, soil organic carbon content in wheat fallow system was nearly 70% less as compared to undisturbed grassland. Therefore, I became interested to understand how soil organic carbon dynamics changes with tillage and nitrogen management practices in dryland winter wheat fallow system. Before going to soil organic carbon dynamics, I would like to start with definition of soil organic matter. Here in this picture we can see crop residues in different states of decomposition, and soil organic carbon include all carbon containing compound or all carbon containing plant and animal residues ranging from very fresh residue to highly decomposed, highly stabilized humidified soil organic material. These soil organic matter in different states of decomposition respond differently to management systems and changes in soil environment. Therefore, when we discuss about soil organic matter dynamics, we can classify soil organic matter into different groups. In this presentation, I will be using three pools approach to classify soil organic matter, in which active pool soil organic matter includes all fraction of organic materials that turns over within a year and supplies nutrient for crops grown in the next season. For example, mineralizable carbon and nitrogen, easily decomposable organic materials, and microbial biomass comes into this fraction. And sometimes people also define this fraction as a light fraction soil organic matter. And slow pool soil organic matter are similar to light fraction soil organic matter, but takes relatively longer time to decompose. For example, it takes several decades to decompose and release nutrients. And some of these organic matters are protected from mineralization within a soil structure. For example, aggregate protected soil organic matter. And another pool of soil organic matter takes centuries to millennia to decompose, and then that fraction is classified as passive pool soil organic matter. For example, some harder tissue structure and some complicated compounds, humidified molecules. So this fraction of soil organic matter is a reservoir of nutrients in the soil system. In undisturbed condition, slow and passive pool soil organic matter constitute major fraction of soil organic matter. But under disturbance, this slow pool soil organic carbon is transformed into active pool soil organic carbon. And then in disturbed systems such as agricultural systems, active pool soil organic carbon is really big, and then this pool can contribute nutrients, but at the same time that is very sensitive to management and easily liable to loss during repeated tillage activities. Therefore, my research objective were to evaluate the effect of tillage and fertilizer nitrogen management on soil organic carbon dynamics in winter wheat summer fallow system. Specifically, I looked at 24-hour soil respiration, potentially mineralizable carbon, microbial biomass carbon, and soil metabolic quotient to understand these system responses to tillage and nitrogen management. We used tillage fertility long-term experiment for this purpose. Then this experiment was established in 1940 in wheat fallow system, and this have three tillage systems and 5 nitrogen rate treatments replicated 3 times. These 3 tillage systems include moldboard plow tillage, offset disc tillage, and soft surface sweep. And these treatments have been modified in several time periods. Specifically, nitrogen rate changed over time. And then from 1988 to 2010, 5 nitrogen rates compared in this study include 0, 45, 90, 135, and 180 kg nitrogen application. Soil samples were collected during summer 2014 from plots that received 0 and 90 kg nitrogen application under each tillage systems, and we measured potentially mineralizable carbon by aerobic incubation method, in which 20 gram soil samples were aerobically incubated in 1 liter jar, and CO2 released during incubation period were collected into syringes and measured into liquor for carbon content, and microbial biomass was determined by fumigation incubation method, and microbial metabolic quotient was determined as a ratio of soil carbon respired during week period to soil microbial biomass carbon. Here comes the result. Example, 24-hour soil respiration was significantly higher under sweep and disc tillage than in the plow tillage system in 0 to 10 centimeter soil depth. And the response of plow tillage on 24-hour soil respiration was higher than disc and sweep tillage in 10 to 20 centimeter soil depth. We did not see any difference in soil respiration in 20 to 30 centimeter soil depth. And in addition, we also observed response of soil nitrogen application, specifically in plow tillage system in 10 to 20 centimeter soil depth. Potentially mineralizable carbon also followed similar trend. Specifically, disc and sweep system have higher potentially mineralizable carbon than plow tillage system in 0 to 10 centimeter soil depth. We also observed a difference in potentially mineralizable carbon content between 2 nitrogen rate treatments in sweep tillage system. In 10 to 20 centimeter soil depth, the response of nitrogen was observed only in plow tillage system, and then potentially mineralizable carbon content was also higher in plow tillage system than under disc and sweep tillage. And the more interesting response of treatments were observed in 20 to 30 centimeter soil depth. I'm not quite sure what is going on in the systems, but plow tillage systems had a higher nitrogen, potentially mineralizable carbon with nitrogen addition, and then potentially mineralizable carbon was decreased with nitrogen addition in disc and sweep tillage system. Soil microbial biomass followed more or less similar trend as 24-hour soil respiration and potentially mineralizable carbon. There was no nitrogen rate response in 0 to 10 centimeter soil depth in any tillage system, and microbial biomass was highest under disc tillage in 0 to 10 centimeter soil depth, and it was higher in plow tillage system than other 2 systems in 10 to 20 centimeter soil depth. Microbial biomass carbon was not significantly different in 20 to 30 centimeter soil depth in soil Soil metabolic quotient showed some interesting trend. This specifically soil metabolic quotient was significantly influenced by both nitrogen application and tillage systems. For example, in top 10 centimeter, it was higher under disc and sweep tillage than under plow tillage, and nitrogen application increased soil metabolic quotient, whereas in lower soil depth, nitrogen application increased metabolic quotient, but the response was higher under plow tillage than under other tillage systems. Example, in 20 to 30 centimeter soil depth, the only difference in nitrogen rate was observed in plow tillage system, but no difference in disc and sweep tillage system. When we looked microbial activity indices in response to soil mineralizable carbon content, we found that 24-hour soil respiration increased linearly with increase in potentially mineralizable carbon content, indicating that 24-hour soil respiration could represent well with potentially mineralizable carbon content. And soil metabolic quotient, which is the ratio of mineralizable carbon with microbial biomass, actually did not increase linearly with mineralizable carbon content, indicating that microbial biomass or microbial response to mineralizable carbon content was constrained by some other environmental factors at higher mineralizable carbon availability. So what can we conclude from this experiment is minimum tillage approaches such as disc and sweep can increase soil organic carbon accrual mainly by improving mineralizable carbon accumulation and with a disproportionate increase in soil metabolic quotient or disproportionate change in microbial activity and potentially accumulation of potentially mineralizable carbon. And soil nitrogen addition also have a similar effect that can enhance the soil microbial activity and increase potentially mineralizable carbon content. But the risk here is that this highly sensitive fraction of soil organic matter can be easily lost under disturbed systems. So repeated tillage can cause loss of this accrued benefit in the systems. With that, I would like to thank you all for your time to go through this presentation, and I would like to thank my research team and Tri Universities and REACH for providing me this opportunity to share my research with you all. Thank you.